This video will cover glycolysis and anaerobic respiration. I recommend that you take notes on your own sheet of paper as I complete the video. Glycolysis is the first step in both aerobic respiration and anaerobic respiration. This video will only cover glycolysis and anaerobic respiration. The other steps of aerobic respiration will be covered in later videos. Aerobic and anaerobic respiration begin with glycolysis, and glycolysis begins with glucose. There are other molecules that can be used as respiratory substrates, but glucose is the main one used by the body. The formula for glucose is C6H12O6, and it is a 6-carbon molecule, which I'm going to represent here by drawing 6 carbons in a row. In actual fact, those 6 carbons are arranged in a ring structure, but we're just going to represent them here as being 6 carbons like so. Glycolysis happens in the cytoplasm, not the mitochondria. So respiration, be it aerobic or anaerobic, always starts in the cytoplasm. The first step is phosphorylation of the glucose molecule. In this step, 2 molecules of ATP are used to phosphorylate the glucose molecule. This produces 2 molecules of ADP. The ATP was hydrolyzed in this process, and the phosphate groups provide energy to the glucose molecule as they phosphorylate it, lowering the activation energy required for the following reactions. This process produces glucose phosphate, which is a 6-carbon molecule with 2 phosphate groups attached. This molecule is then broken down into 2 molecules of triose phosphate. These are 3-carbon molecules with a phosphate group attached. From now on, everything is doubled because we have 2 triose phosphate molecules. The first thing that happens to each of these is that they are oxidized. That means they lose an electron. The electrons are picked up by high energy electron carriers called NAD+. This then forms NADH, or reduced NAD. As we have 2 triose phosphate molecules, this process happens to each of them, creating 2 molecules of NADH. NADH is a high energy electron carrier, much like NADPH that was found in photosynthesis. In aerobic respiration, the NADH will go to the electron transport chain inside mitochondria and provide it with high energy electrons to power the production of ATP. So, our triose phosphate was oxidized when it lost the electrons. Then, the molecules are used in the production of ATP from ADP and PI. This happens twice to each of our 3 carbon molecules. So let's draw that in. This means that the cell is gaining energy from the breakdown of glucose. Think about it, that's what glycolysis means, sugar splitting. This is called substrate level phosphorylation. At the end of this process, we are left with 2 3-carbon molecules. These 3 carbon molecules are called pyruvate. The pyruvate still contains a lot of the energy that was in the original glucose. Pyruvate will also move to the mitochondria if there is oxygen present. In the mitochondria, pyruvate will be used in the link reaction, which will follow on to the Krebs cycle and then the electron transport chain, if there is oxygen present. The NADH, ATP, and pyruvate are the products of glycolysis. Let's think about energy inputs and outputs. To start this process, we had to put energy in the form of 2 ATPs. So there was a total input of 2 ATPs at the beginning. The end of this process gave us an output of 4 ATPs. That means there was a net profit of 2 ATPs. That's not a lot of energy. Glycolysis releases some, but not much energy from each glucose molecule. Certainly not much compared to the amount of ATP produced by aerobic respiration when we have mitochondria involved as well. This pyruvate has a lot more energy left in it. 2 molecules of NADH are also being produced in glycolysis, along with our 2 molecules of pyruvate. These 2, the NADH and pyruvate, still contain a lot of energy, which will only be released if oxygen is present. So let's talk about what happens when oxygen is not present. Anaerobic respiration. Remember, anaerobic respiration includes this whole process. This is glycolysis. But anaerobic respiration happens if there is no oxygen present. For reasons that will be discussed in a later video, if no oxygen is present, this pyruvate and NADH have nowhere to go. This creates a problem because the cell ultimately will run out of NAD+. If all of it has been converted to NADH, it has been reduced, forming reduced NAD, then there will be no NAD+ left for the cell to keep this process going. Glycolysis can be really critical if you need that little bit of extra energy when there's no oxygen available. So your cells need to replenish the NAD+ here so that this can keep going. To do that, cells use a process called fermentation. In animals, fermentation results in a product called lactate or lactic acid. The purpose of fermentation is to oxidize the NADH so that it returns to its form of NAD+. In other words, the pyruvate is accepting electrons from the NADH, and it is itself becoming reduced to lactate. That will provide the NAD+ needed to keep this process going. Lactate is produced by animals, but that is not the case for some other groups of organisms. In plants and fungi and some bacteria, the products are different. Their fermentation results in the production of ethanol and carbon dioxide. Notice carbon dioxide is not produced when lactate is made in animals. So this happens in plants and fungi such as yeast. It also happens in a few bacteria. In animals, this would be really bad news because ethanol would interfere with our immune system. Sorry, not our immune system, our nervous system. In plants and fungi, this is filling the same purpose of replenishing NAD+ by oxidizing the NADH, by taking the electrons from it. So, to summarize, this central process here, this is glycolysis, the splitting of sugar. In aerobic respiration, pyruvate and NADH will go to the mitochondria. But in anaerobic respiration, when there is no oxygen present, the pyruvate is fermented, creating either lactate in animals or ethanol and CO2 in plants and fungi. The purpose of that is to regenerate the NAD+ so that this process can keep going, and so that that net profit of 2 ATP can continue to be released. One other thing that I'll mention that is not on the A-level spec but is worth knowing, I believe, is that the enzymes involved in this pathway have some really important regulatory functions. The first enzyme involved in this is called phosphofructokinase, and the reason I think it's a really interesting enzyme is that it can be sped up or slowed down by the presence of ATP and ADP. If ADP is present, then phosphofructokinase accelerates this process. The enzyme works faster. Think about how useful that is. The enzyme is working faster because the cell needs more ATP. It needs more ATP to be produced. It needs respiration to happen faster. If there's a lot of ADP present, it suggests that the cell is already working hard, using a lot of energy. Amazingly, this same enzyme is inhibited by the presence of ATP. So if a cell has an abundance of ATP, respiration is stopped because ATP inhibits the first enzyme in the process. A beautiful mechanism for regulating the rate of respiration in cells, and so the rate that ATP is being produced. Why break down the glucose, ultimately, if it isn't needed by the cell, if the cell has plenty of ATP already? To summarize again, this is glycolysis. It can be followed by either the rest of aerobic respiration or fermentation. This whole thing together would be what we refer to as anaerobic respiration. I hope you found this video helpful.